Bismillahirrahmanirrahim. This is the 8th video in the series of molecular basis of cancer. In this video, we will discuss angiogenesis. These are the references for this video. In homeostasis, there is a fine balance between forces that favor new blood vessel formation and the forces against the new blood vessel formation. So the endothelial cell turn over slowly, take about months to years to renew themselves. The tumor disrupt this equilibrium in favor of new blood vessel formation. This is termed as phenomenon of angiogenesis. So this angiogenesis can be defined as the formation of new blood vessels from pre-existing small blood vessels, and this is obligatory for a primary tumor to grow and metastasize. The mechanism for the development of or formation of these new blood vessels is due to hypoxia, which is induced by the rapidly growing tumor, and there is induction of hypoxia induced factor HIF1 alpha as a result of decreased blood supply in the tumor cell, and these will in turn activate the angiogenic growth factors that are responsible for the formation of new tumor associated blood vessel, and the new tumor blood vessels are as necessary for the tumor growth, and they allow tumor to grow beyond about 2 nm in diameter. The formation of the new blood vessel starts as the tumor cell or the stromal cell, they secrete various angiogenic substances. The steps in the formation of new blood vessels are: first, proteolytic enzymes are secreted that perforate post-capillary venule basement membrane. The endothelial cells in the area of interrupted basement membrane proliferate, and they migrate toward the source of angiogenic cytokine. This advancing mass develops a lumen within it, and this tube-like structure is then invested by a basement membrane, and the advancing part of this developing structure, that is the tip cell, join the other similar tip cell, and hence a capillary network is formed. For some tumor types, the tumor vessels may form directly from the tumor cell themselves. So it is possible to differentiate the tumor associated vessels and the non-tumor associated vessels because they differ in various characteristics. One is that tumor associated capillaries may be variably invested by pericytes and basement membrane. They may vary in size and shape. They leak blood component excessively. They display excessive tortuosity, and they are distributed inhomogeneously within the tumor mass. As they are excessively leaky, there may be increased tissue hydrostatic pressure, so the diffusion of soluble material into the tissue is impeded. So some areas of the tumor may be richly supplied with oxygen and nutrients, while some are not. There are three main groups of the mediators of tumor angiogenesis. First is the family of vascular endothelial growth factor. Second is the angiopoietins, and the third is the group of other agents that stimulate tumor associated blood vessel to proliferate. These are transforming growth factor beta, several interleukins such as interleukin 6 and 8, and steroid sex hormones. The vascular endothelial growth factor is a family of related protein. The common is the vascular endothelial growth factor A that has got several isoforms, but there are many vascular endothelial growth factor which are interrelated, and the common source is tumor cell, while other sources are platelet and connective tissue cell. There is another important factor, that is platelet derived growth factor, and this has got similar spectrum of activities to the vascular endothelial growth factor. The functions of VEGF are stimulation of new blood vessel formation, capillary permeability, it promotes endothelial cell survival, and also mobilizes progenitor cells from the bone marrow. When there is hypoxia, that means insufficient oxygen, that will result in transcription factor formation, that is hypoxia inducible factor 1 alpha. This transcription factor upregulates diverse genes including VEGF gene, and the VEGF binds a family of receptors on endothelial cells. These are VEGFR2, and this ligand receptor interaction will result in activation of signaling pathway. When this signaling pathway is activated, there will be endothelial cell proliferation, protection from programmed cell death, enhanced cell migration into that area, and increase of vascular permeability. When there is increased vascular permeability, the blood component leak into the area, and fibrinogen, especially outside the blood vessel, it generates a fibrin matrix that facilitate endothelial cell migration and angiogenesis. The VEGFR2 positive progenitors from the blood are also recruited to the site, and these VEGF families are also responsible for the proliferation of lymphatic vessels. So, to summarize the events, we can say that the increased VEGF is a result of either tumor cell factors or extra tumor factors. Increased oncogenes such as EGFR, RAS, CSRC, etc., and decreased tumor suppressors activity such as P53 and P10 will result in increased concentration of vascular endothelial growth factor. The extra tumor factors that are responsible for increased concentration of VEGFs are hypoxia, chemokines, cytokines, growth factors, and other soluble mediators. As a result of increased concentration of VEGF, it binds and activates VEGF receptors. There is a family of VEGF and also different receptors such as VEGFR1, receptor 2 and receptor 3. The principal among these is VEGFR2 that binds with the VEGF-A's, and when receptor ligand interaction occurs, it activates the intracellular signaling that will result in increased vascular permeability, proliferation, increased survival of the endothelial cell, migration of the endothelial cell, and mobilization of the endothelial cell progenitor. Several angiopoietins have been recognized. The angiopoietin 2 contribute the most to the tumor blood vessel formation. It stabilizes growing blood vessel and stimulates pericyte to surround the developing structures. So after recognition of the VEGF and its receptors, scientists thought that we can stop the tumor growth by blocking these VEGF or its receptor. Hence we can block the downstream signaling pathway, but there are many other factors and mechanisms that are involved in these angiogenesis, and these are the cytokines such as interleukin-6, androgen and estrogen, and other growth factors that have got diverse functions, including evoking VEGF production. Then mutation in several important oncogenes such as SRC, EGFR and RAS, and impairment of activities of certain tumor suppressor gene will result in increased VEGF secretion by tumor cell, and there is multiplicity of the proangiogenic activities include such factor as basic fibroblast growth factor, platelet derived growth factor, VEGF homologues and isoforms, and many others. These may augment blood vessel growth that is triggered by primary factors such as FGF2 or angiopoietin. The soluble angiogenic factors and stimulators may also be produced by some immune cells and inflammatory cells, including macrophages, neutrophils, NK cells, dendritic cells, and myeloid precursor cells. Some cells even have tendency to differentiate into endothelial cells. The role of tumor-associated stromal fibroblast is not fixed. In some settings, it assumes an anti-neoplastic phenotype and produce anti-tumor and anti-angiogenic activities. In other settings, they generate pro-angiogenic and pro-tumor microenvironment. The dendritic cell and myeloid-derived suppressor cells are also capable of trans-differentiating into endothelial cells. So by this discussion we can infer that diverse population of cells, including tumor cells and bone marrow derived cells, participate in angiogenesis. These are especially the tumor cells, the stromal cells, the macrophages, the neutrophils in the early myeloid series, and myeloid derived suppressor cells, endothelial progenitor cells, etc. To end the discussion on angiogenesis, we can say that a solid tumor cannot grow beyond 1 to 2 mm in diameter unless the tumor has the capacity to induce angiogenesis. The health of the tumor requires delivery of oxygen and nutrient as well as removal of the waste product. Hence, angiogenesis is essential for the tumor to grow. This is stimulated by growing cancer. These new blood vessels fulfill the function of nutrient and oxygen supply, but also stimulate the growth of adjacent tumor cell by supplying growth factors such as insulin-like growth factors and platelet-derived growth factor. These new channels are leaky and dilated, and they have a haphazard pattern of connection that can be visualized on angiographs. The angiogenesis also contribute to metastasis by permitting tumor cells ready access to these abnormal vessels. The angiogenesis is controlled by a balance between promoter of angiogenesis and its inhibitor. In angiogenic tumor, there is imbalance in favor of angiogenic promoter. The angiogenesis is not induced in the start of the tumor development, and tumor remains small and quiescent, and this stage ends when angiogenesis is induced, and this is the signal that favors tumor progression. This achievement of the induction of angiogenesis is through increased local production of angiogenic factor or loss of angiogenic inhibitor, and these factors are derived from tumor cells, the inflammatory cells that have been infiltrated into the tumor, such as macrophages and other cells that we have just discussed, and Other tumor-associated stromal cells are also responsible for these factors, and some of the factors are also derived from extracellular matrix. In the case of tumor cells, several alterations enhance the production of proangiogenic factors. The lack of oxygen will result in stabilization of hypoxia inducible factor, that is oxygen sensitive transcription factor, and it activates the transcription of proangiogenic cytokines such as vascular endothelial growth factor and basic fibroblast growth factor. This leads to development of an angiogenic gradient, and it stimulates the proliferation of endothelial cell and guides the growth of new vessels toward the tumor. The driver mutations in certain tumor suppressors and oncogenes favor angiogenesis, as P53 controls thrombospondin, that is anti-angiogenic. It also controls the VEGF. So the loss of P53 will result in deletion of repression of this VEGF. So VEGF is expressed more. Also, the RAS and MYC, if they get gain of function mutation, the vascular endothelial growth factor will be released more. The proteases are also involved in this play. They are elaborated either by tumor cell or by stromal cell, and they also disturb the balance of angiogenic and anti-angiogenic factors. Many proteases release pre-absorbed basic fibroblast growth factor from the extracellular matrix, while some proteases, they release anti-angiogenic factors such as angiostatin and endostatin. Angiostatin is the product of proteolytic cleavage of plasminogen, while endostatin is the product of cleavage of collagen. The angiogenesis is essential for solid tumor to grow to achieve a clinically significant size, and this provided a clue for the development of therapeutic agents that block angiogenesis. Bevacizumab is an example that is a costly drug and adds few months to victim's life. This is a monoclonal antibody that neutralizes vascular endothelial growth factor activity, and it is approved as a treatment of many tumors.